Today we're going to continue our discussion about T cells in the periphery. Last time we talked in detail about this original interaction where our T cell is interacting with an antigen-presenting cell. That antigen-presenting cell is, as its name suggests, presenting antigen to stimulate that T cell through its T cell receptor and lead to a signal transduction cascade. And in the end, our T cell is going to start producing IL-2. We then have some additional steps that are going to happen with our T cell before that T cell can go ahead and induce its effector function. And I will just say, if I haven't clearly defined this before, effector function is a term that immunologists love. It means doing your job or doing whatever your effect is. We use it all the time. So a killer T cell's effector function is killing, and a helper T cell's effector function is making cytokines. And when I was first practicing a talk for interviewing for a job, my friends started yelling at me because they're like, you're using the word effector function all the time, and people who aren't immunologists don't know that word. And I was like, what? What do you mean somebody doesn't know that word? How do people not know that word? So that's when I learned people didn't know that word. And now that's what it means if I use it again here. But as I told you last time, the real details of activation of T cells involve additional signals. So last time we talked about this signal that's labeled here in your textbook with a number 1 next to it, and it is very frequently called signal 1. So we just saw a lot about how signal 1 works when we activate T cells. But in real life, there are additional T cells, or additional signals, the most important of which is one that's usually called signal 2. It's listed here as co-stimulatory interaction. And there are also some important cytokine interactions. Some people refer to those as signal 3. So, to fully activate a T cell, and this is specific to fully activating a T cell, when it is a naive T cell, activating that T cell for the first time. The T cell, in fact, requires the receipt of 2 signals, not just 1. So we talked about that whole signaling process last time. That's all signal 1. Our T cell also has a protein on its surface called CD8. And not only does our T cell need to get a signal through its T cell receptor in order to be activated, that T cell also needs to get a signal through CD8 to be activated. CD8 has 2 possible ligands that are shown here. Those ligands are listed as CD80 and CD86. The other names for CD80 and CD86 are B71 and B72, or just B7. So sometimes you might see B7 listed. B7 is the same thing as CD80 or CD86. I'm not really going to say anything that's special about one versus the other. As far as I'm concerned, they can be CD80 and 86, or they can be B7 or whatever. But those proteins are produced by our antigen-presenting cell. They're transmembrane proteins on our antigen-presenting cell that can bind to CD8 to give this second signal to our T cell. And this process is critical for our T cell. A T cell must receive signal 1 and signal 2 in order to become activated. If a T cell receives signal 1, as is shown here, without signal 2, and again, this is a naive T cell that is being activated for the first time, that T cell does not become activated. But it's not just that. Not only does the T cell not become activated, that T cell is actively turned off. That T cell is energized. So if signal 1 and signal 2 are provided together, our T cell is activated. If signal 1 is provided without signal 2, our T cell is energized. if the T cell were to get signal 2 without signal 1, which is a question people sometimes ask, this is exceptionally important. This is really what makes this whole T cell process work. So there are a lot of parts that we can talk about in terms of how this is important or what goes on. And so the first thing that I want to talk about is how does this antigen presenting cell know to make its co-stimulatory signal, its B7 or CD80 or CD86, whatever we're going to call it. How come this antigen presenting cell knows to make a second signal and turn on the T cell, and this one doesn't turn on B7, CD80, CD86 to turn on the T cell? How does the antigen presenting cell make that distinction? And the answer is pattern recognition. So one of the other effects of the pattern recognition receptors that we saw before is that their signal transduction turns on the production of co-stimulatory molecules. And in fact, this sort of insight is what we're talking about with those papers on Monday of next week. That's why we're tying those papers in here. And so there's another way that you could look at this. When I talked about T cell development, I told you about this problem in the thymus. And if I remember correctly, you asked questions like, how are you still a person and how are we all not dead, after I discussed this information. I told you that a thymocyte that develops in the thymus with high affinity for self-antigen will undergo apoptosis through negative selection. And I told you that a thymocyte with low affinity for self-antigen will develop into a T cell. And we learned that every T cell that develops has low affinity for self-antigen. Every T cell is a little bit self-reactive. And again, you all thought we were going to die because of this. And it turns out the answer to some of the questions that you wanted to know at this moment actually are signal 2. The reason why this works is because of this process called cross-reactivity. So we have lots of self-proteins that are very similar to the epitopes from different pathogens. And so if the T cell binds weakly to that self-protein, they could potentially bind strongly to the similar non-self protein. Maybe the 1 or 2 amino acids that are different are going to be enough to give strong binding in the periphery. And so you can see lots of examples of such potentially cross-reactive proteins here. You can see, for example, that this insulin receptor has 6 amino acids in common with a protein from rabies. And so a T cell that gets selected with low affinity for this insulin receptor might have high affinity for the rabies protein. So this is one reason why this whole process might make sense and might work. But there's still a lot of danger involved. You can still imagine, well, yeah, but why doesn't that T cell make a response to the insulin receptor and give me diabetes or something like that? And this all comes back to signal 2 and T cell anergy. If our epitope that is being presented on the MHC molecule is an epitope that has come from a microbe, if that it's an epitope that's come from infection in our antigen presenting cell, that antigen presenting cell has the epitope to present, but also has received some type of pattern recognition receptor stimulation through a MAMP, PAMP, DAMP, whatever we're calling it today, and thus has received a signal to make CD80 or CD86. There are a couple of terms that have gone in and out throughout the history of immunology. I'm going to use a term now that many immunologists don't like, and so I'm going to say it because it makes a lot of sense for us in terms of understanding this concept. But when I say it, I always am afraid that like lightning's going to strike me down. So you can imagine that that pathogen product can sort of be like a danger signal. It's telling this antigen-presenting cell, the thing that you have is dangerous. Now, any antigens you're presenting, they are being presented in the context of danger to the cell. There was a microbe around. We saw a microbe, something serious is going on here. And so this antigen-presenting cell, because it has been in the presence of a pathogen, is now going to make signal 2 and actually turn on the T cell because we've made our danger signal. However, if we have another cell that's presenting self-antigen, that cell has not been in the presence of a pathogen. That cell will not be making that second signal, that quote-unquote danger signal. This is sort of indicative of presentation of a self-antigen. And so if our T cell goes into the periphery and recognizes a self-antigen, it's going to do that without the second co-stimulatory signal. This is the case where that T cell could potentially be getting turned on in an autoreactive fashion. And so we don't want the T cell to be turned on, first of all, but we also want to now induce some kind of tolerance. And so the T cell gets shifted to anergy. This is going to be one of our peripheral tolerance mechanisms that we can see. And so this is a really critical part of turning on T cells and also of making sure that we don't have self-reactive responses. This is also very important for us as experimentalists. If we have T cells in a dish and we want to turn them on, we have to make sure we give them both signal 1 and signal 2, even in a dish. And so, for example, we can give cells a drug called PMA that basically induces a signaling cascade like the signal 1 cascade. And it doesn't turn on T cell proliferation very well. We can also give anti-CD3, which just gives us a T cell receptor stimulus, gives decent stimulation. But if we add stimulation through CD28 to either of those in our situation in a dish, you can see we get much better T cell stimulation. And so whether we're talking about an in vivo experiment or an in vitro experiment, we always need to give our T cells both signal 1 and signal 2 in order to fully activate them. So this is sort of the basic view of co-stimulation. In real life, there are tons of co-stimulators on T cells. This is a table from your textbook, and it only shows 5, although it shows 5 that are really important. This is a figure from a paper, and it shows way more than 5. And in fact, it's still an understatement of how many different T cell co-stimulatory molecules there are. There are 2 that I want to tell you about specifically that are described here, in addition to the CD20, CD80, CD86 interaction that I've already told you about. One thing that you can notice on the picture version, it's also actually part of the table, the way they organize the table, is that there's some different colors in this part of the table. One of them is green. The other one is red. When you think about things being colored either red or green, what do you usually think about? Stop, go. Exactly. And in fact, some of these co-stimulatory molecules are activating. They make the cell go. Other co-stimulatory molecules are inhibitory. They make the cell stop. Your textbook just says co-stim receptors, co-inhibitory receptors. Here there's pluses and minuses that I'm not sure whether you can see, and stop and goes. So we've got some co-stim receptors that turn cells on, some that turn cells off. The ones that turn cells on have somewhere in their cytoplasmic domain an ITAM. the ones that turn cells off, have something in their cytoplasmic domain called an ITIM. ITAM is immunotyrosine-based activation motif. ITIM is immunotyrosine-based inhibitory motif. And we'll see a lot more about ITIMs a little bit later. But there are two negative receptors, that both have ITIMs, that I want to talk about a little bit here. And to really describe why they are so important, I'm going to show you this slide. You're actually going to see basically this slide again a little bit later, which is just one quick thing that I want to tell you about the kinetics of a T cell response. And this is true of a B cell response too, but I'm really talking about T cells right there. What you will notice is that at the point of infection, it takes a little while for our response to come up. You can see that our response does come up pretty nicely to some level, but eventually that our response, in fact, comes back down. That's known as the contraction phase, or AICD, which stands for activation-induced cell death. And in my PhD lab, we actually spent a lot of time studying that contraction phase, because what we wanted to do is we wanted to make it go away. We thought that if we were making a vaccine, wouldn't it be cool if we could make a vaccine that gave you a lot of T cells and then they stayed high forever and ever? And they never contracted. Wouldn't that make a vaccine way better? And so we did everything we could think of to block contraction. And the answer is you kind of can't. That contraction phase is super, super, super important. There are lots of redundant mechanisms to keep contraction going. And if you think about it, that makes sense. If you made an immune response to the flu that you had when you were 2 years old, and you expanded T cells, and then you expanded T cells to chickenpox that you had when you were 3, and you expanded T cells to some cold that you had when you were 4, and you add up all the times you increase T cells. By this point of your life, you would be a walking lymph node. You don't have enough room or enough resources to support those huge expanded numbers of T cells to every pathogen you've ever seen. You can see that you keep a higher number than you started with, but you can't keep this huge number. And in fact, whenever you have that hugely expanded number of T cells, you're in a little bit of danger of some kind of self-reactivity, some type of excess pathology, some type of stuff going wrong here. And so the turning off part of T cells is actually as important as the turning on part. If your T cells stayed on forever, you would be in bad shape. Thus, we have important molecules involved in this co-stimulatory process that serve as off switches for our T cells. The most famous of these off switches is known as CTLA4. CTLA4 is really interesting. So CTLA4, like any of these proteins, has a ligand. In fact, CTLA4 has 2 ligands. CTLA4's ligands are B7-1 and B7-2, or CD80 and CD86. Have you ever heard of those before? Yes, when? Like 10 minutes ago, right? Those are in fact the same proteins that bind CD28. And so what happens is at the beginning, our T cell expresses CD28, and our T cell can receive signal 2, and our T cell will get turned on. Once that T cell has been sufficiently activated, it will start making CTLA4 in addition to CD28. CTLA4 has higher binding affinity to B7-1 and B7-2 than does CD28. And so in a situation like the one you see on this slide, CTLA4 will win. It will steal all the B7-1 and B7-2. CD28 will no longer bind to the B7s. And this will lead to a stop signal in the cells. And so our cell will now be turned off and inhibited. And so this is a pretty nice system. Basically, as soon as our cell gets turned on, it immediately also puts an off switch on its surface so that it can be turned off and we don't have to worry about excess activation. And you can see this process happening here as well. That our naive T cell, shortly after activation, will start making CTLA4 in order to make sure that it is a controllable T cell, that its response can be controlled. There is another protein that is similar to CTLA4, known as PD-1. CTLA4 is shown at the top of this slide, and the T cell that's shown in the top version is a naive T cell or a resting T cell. It's a T cell that's early in its life, early in being activated. If that T cell is more of a memory T cell, an antigen experienced T cell, it might not have CTLA4, but it might instead have a negative protein called PD-1, which is able to bind to one of two ligands called PD-L1 or PD-L2. You can see that here. So it's basically the same process, just that process is now happening on an antigen-experienced cell instead of on a naive cell. So, the question to ask you about. Let's imagine that you were a T cell that responded to influenza. I'm going to go back for a second. Okay, you're a T cell that responds to influenza. You're this influenza T cell. And we look at this plot, and this is the timing. What's going on with the influenza virus during this time period? Is there a lot or a little influenza virus right here? Yeah, Jordan. It's replicating. There's probably a lot of it replicating here. What's happening to it here?
It's hopefully dying.
It's hopefully dying. It's hopefully gone. And I picked influenza because it is a virus that gets cleared. So once you have a good T cell response, that virus is gone. It is cleared from your body. There is zero of it left. And so if you are a T cell at this point, influenza is gone. Really, sort of it throughout this point. And so what are you going to do as that T cell at this point? Some of them are going to die. What if you're one that doesn't die? What are you going to do? Nope. What are you going to do, Molly?
I'm going to, rest.
Yeah. Now it's time to put up your feet and lay down and take a nap. That's where that T cell is going to now be able to go back to being a resting T cell. That T cell is sort of going to get a break. If you imagine now the same scenario with HIV, what's the problem? Yes, Kay.
They're never going to break.
You know, the T cell's never going to break. HIV is never cleared from an infected person. Those people are going to be responding to HIV for the rest of their lives. You can also think about, and this might be something we'll come back to, so I'm only going to mention this a teensy bit right now, about how self-antigens are also present for long periods of time. Also, really importantly, tumor antigens are present for a long period of time. And so if you're that T cell that's responding to HIV, maybe a self-antigen, but that's a whole argument, or a tumor antigen, what are you going to do here? Or are you ever going to lay down and take a nap in your hammock? This is my hammock. Any hammock time for you? No hammock time. Your virus is never gone. So you never get to relax. If you never get to relax, and you know about this from this time of the semester, if you never get to relax, what happens to you? What happens to you, Molly?
Exhaustion.
You get exhausted. I heard Alina say you get stressed and die. It's the same thing as exhaustion. And in fact, when antigens are present for long periods of time, the T cells are dysfunctional because of something known as chronic exhaustion. They stop making their cytokines, they stop having their ability to kill. As you can see, over time, we get more and more minuses. We have the cells lose more and more of their ability to do these processes. And if the cell gets really exhausted, it's going to undergo apoptosis as it got stressed and died, like Alina said. Yep.
So, I imagine this isn't really a case that happens often, but if you were started to have dysfunction, and then some reason you got cured, would your immune system still be dysfunctional, or would they be able to rest and recover?
So that's a great question. And so we don't totally know the answer to that question. But I'm going to tell you a couple things on this slide and the next couple slides, and then we'll see what we hypothesize is the answer, because nobody has really done that. One of the things that happens when these cells become exhausted is they start making huge amounts of PD-1, that negative signaling molecule. They start making huge amounts of CTLA4. It's not shown here, but they start making huge amounts of those negative signaling molecules. That's part of exhaustion. Those cells are going to get turned off really easily. The Nobel Prize this year was awarded to two scientists who were dealing with these exact molecules. So one of them was a person named Jim Allison, who came up with his first idea of checkpoint inhibition. Basically what he did is he designed an antibody against CTLA4 so that CTLA4 is blocked and we can never turn off a T cell. The T cell cannot actually ever get that negative signal through CTLA4. It's blocked, so B7 always binds to CD28. This can be useful in tumor patients, where T cells stop killing tumors because those T cells get exhausted. So now we stop the T cells from getting exhausted by blocking this molecule that's involved in the exhaustion signal in CTLA4, and the T cells live happily ever after and are persistently activated. I'll get to you in just a second, Molly. So one of the two people who won the Nobel Prize did this. The other one designed basically the same molecule, but it was against PD-1. You may have heard of it, of pembrolizumab, Keytruda, or Opdivo. It's the same thing, it's just blocking PD-1. It's largely being used in cancer patients to block T cell exhaustion. So you can see our exhausted T cell is getting some signaling through PD-1. When we treat with this antibody, so PD-1 can no longer make its negative signal, it's reinvigorated, as this review says. This is also being used in some situations where our patients are having problems with T cell exhaustion, and there are ideas of using particularly the PD-1 drugs in HIV patients, because PD-1 exhaustion is a pretty big deal with HIV patients. So based on the efficacy of these things, if you could reverse the exhaustion, you'd be okay, but you'd have to figure out a way to reverse the exhaustion, but that's based on the effects of these drugs. There isn't another kind of clear experiment that somebody has done. Molly, what's your question?
These aren't specific for T cells, right? Like for specific T cells.
No. So in fact, so why is that a problem, Molly?
Autoimmunity. Also, if you have cancer and you get the flu, you won't ever, those T cells won't stop proliferating or signaling, so you get cytokine storms.
Yeah, so you're basically blocking all of the negative signaling. You're blocking all of the CTLA4, all of the PD-1 in your immune system. You're not doing it in a tumor cell specific way here. And so, this is better than totally killing your whole immune system. If you use a CTLA4 blockade, for example, you still have PD-1, so your T cells can sort of get turned off. So it's better than sort of being too broad, but you're right that these do still have the problem of we wish they were a little more specific. You can certainly imagine potential autoreactivity side effects that could happen, but if you're a cancer patient who doesn't have, other things that are going to work for them, this can be really helpful. Yep.
So could this work is there a half-life of the drug? Is it just
Yeah, these are antibodies.
Yeah.
And so there is a half-life. So you usually have to have repeated administrations of them. And you're going to administer them until you've gotten rid of the disease, and then you're going to stop administering them. So yes, there would be half-life. So this is how this whole signal 1, signal 2 business became really hot in the news this year. We also have this an issue of cytokines being really important in further helping those T cells become activated, and particularly in terms of helping those T cells fully differentiate. So helping the T cell obtain its adult function, attain its effector function. So as I told you last time, a naive CD8 cell isn't going to be a killer. Only an activated CD8 cell is going to be a killer. This helps that differentiation process. The cytokine that I'm going to talk about today is IL-2. You'll see some other cytokines on Wednesday. And IL-2 was happily the cytokine that we saw on Monday that was produced by our T cells in response to turning on the transcription factors NFAT, AP1, and NF-kappa B. When IL-2 was first cloned, it was named T cell growth factor, TCGF, which gives you a good hint about what IL-2 does. IL-2, when it signals to on our T cell, when it signals through its IL-2 receptor, will push that T cell into cell cycle and allow for many different divisions of that T cell, as well as allowing that T cell to gain its effector function. And so you can see we've got proliferation and differentiation happening because of IL-2. IL-2 is a member of a family of cytokines. I always feel a little funny doing this slide because I worked on a member of this family of cytokines for my PhD. So I have a figure like this in my dissertation and in my dissertation talk, and I have to tell myself, no, you can't go. You can't do that talk now. You got to do this talk. So there are a member of cytokines, and the reason why these cytokines are classified as a family is that they all have these receptors that have multiple parts, and one of the parts is shared among all of them. So they all use this one chain, the gamma chain, known as the common gamma chain, in their receptor, and pair it with a different protein in order to make that receptor. And so the cytokines of the common gamma chain family are IL-2, IL-4, IL-7, IL-9, IL-15, and the one I worked on that's not shown here, IL-21. So they all signal through the common gamma chain. And in fact, the most common genetic immunodeficiency that people think of when they think about big-time genetic immunodeficiencies is actually a defect in the common gamma chain, which knocks out all of these cytokine responses. That includes IL-7, which you've seen before, which is super important for B cell and T cell development. So you don't have this cytokine chain, the cytokine receptor chain, you have no B cells, no T cells, your NK cells don't work. Basically, your whole adaptive immune system and parts of your innate immune system are gone. So the IL-2 receptor uses the common gamma chain. The IL-2 receptor is a little bit unique from some of the others because it pairs 2 other chains with gamma instead of just one. So there's also the IL-2 receptor beta chain and the IL-2 receptor alpha chain. The common gamma chain is known as CD132. IL-2 receptor beta is known as CD122. And the one of these you actually care about is IL-2 receptor alpha, which is also known as CD25. And so we are going to see CD25 pop up here and there. It's IL-2 receptor alpha. All 3 of the different forms of IL-2 receptor that can be made with these proteins can bind to the cytokine IL-2, but they bind with different affinities. So if we just have IL-2 receptor alpha, there is a very low affinity for IL-2. If we just have the common gamma chain and beta, we have intermediate affinity for IL-2. And when all 3 chains are together, then we get very high affinity binding to IL-2. So we can have differences in that receptor binding. At the beginning, when our T cells are naive, they express only the beta chain and the gamma chain and have a moderate affinity for IL-2. So they can get some signals from IL-2. When that T cell starts to become activated, that T cell will begin to make the IL-2 receptor alpha chain in addition, and will suddenly have a high affinity IL-2 receptor to bind to IL-2 much more strongly than it could before. Once this binding occurs, we will get signaling through the IL-2 receptor. On the next slide, I'm going to tell you about cytokine signaling in general, because again, this is one people like. Of all the types of signaling one has to memorize, cytokine signaling is one people usually get pretty easily. We're specifically talking about it now because of it being the type of signaling that happens with IL-2 receptor, but in fact, you can see similar signaling with most every cytokine receptor that you have. So when we think about cytokine receptor signaling, our cytokine receptors pretty much always have two different chains, and they diffuse separately in the membrane. In the case of IL-2, it might be 3 chains, but they're diffusing separately in the membrane. Once the ligand, the cytokine, is present, that induces those two chains to come into proximity with one another. So the extracellular domains are brought close to one another, which of course brings the intracellular domains close to one another. The intracellular domains of the cytokine receptor are bound to proteins known as JAKs. JAK can stand for a couple things. It's so exciting that it made me roll it over. So JAKs have a couple different parts. So JAKs have actually 3 important pieces to them. They have this one area that's a binding site. They have a tyrosine that's pretty important, and they also, you kind of can't really see the way you draw it like this, but it also is a kinase down here. And when 2 JAKs are brought close together, they can phosphorylate one another. Can I draw this? We all know the answer is no. So they will phosphorylate one another on those tyrosines and be able to then bind the phosphorylated tyrosines to the binding site, so they'll kind of make this back-to-back structure. And you can see that somewhat here as well. So the JAKs are sometimes thought of as these kinases that have 2 faces. They have the 2 different parts that allow them to make a complex. And so the name JAK came from Janus kinase. Janus is a Roman god who has 2 faces. Or maybe a Greek god. I don't know, one of those. I should learn that before Jeopardy. It's Greek?
It's Greek.
See, I got one.
Percy Jackson, that's why.
Okay. So anyway, Janus, 2 faces. So the idea is that the kinase has 2 faces that allow both phosphorylation and binding, and can give you something that looks like this. I am told by old school immunologists that that is a lie. That they first found JAKs in some patients who had immunodeficiencies. They also were deficient just like the patients without common gamma chain, so they didn't make some type of T cells and B cells. And they got super excited when they found them. And they said, oh my gosh, we found the answer to how T cells and B cells work. We're so psyched. And then they kept doing their studies, and they found out they had found just another kinase. And so if you ask people who are in the field a while ago, they tell you that JAK stands for just another kinase. And that's only later on did somebody make up the fancy name of Janus kinase. There are 4 different JAKs, JAK1, JAK2, JAK3, and TYK2, because of course it can't be simple. And different cytokine receptors will bind to different specific JAKs. Sometimes there'll be pairs of different ones. Sometimes they'll be sort of specific. So which individual JAK is being turned on is sort of important here. Here we're just saying it as JAK. And then the JAKs also were able to phosphorylate other proteins. The other proteins that are phosphorylated are known as stats. You can see the stats getting phosphorylated here, and you can see that the stats are able to form dimers similar to those formed by the JAKs. There are, again, a few different stats, so 1, 2, 3, 4, 5A, 5B, and 6, I think. So there are a bunch of them that come together, and STAT stands for signal, transducer, and activator of transcription. And so once the stats are activated, once they're dimerized, and again, whether it's a dimer of like STAT5 with STAT5 or whether it's STAT1 with STAT5, will be unique for different cytokines. Once our stats are dimerized, they actually are themselves able to be transcription factors. So the stat is now able to go into the nucleus, specifically bind DNA and activate transcription. That's why it's important that the AT is the activator of transcription. So the cytokine signaling pathway is usually known as the JAK-STAT pathway. And if you remember the name of this pathway is the JAK-STAT pathway, you know all the names of all the members. And you even know what order they work in. So usually this one's a pretty easy one for people to remember. So what will happen with our T cells is that our T cells at the beginning will express the low affinity IL-2 receptor. Once that T cell becomes activated, the T cell will start making IL-2 and will also start making the final chain, the alpha chain of the IL-2 receptor. The T cell can then actually stimulate itself via the IL-2 receptor to induce proliferation of that T cell. Usually this is happening in sort of a local area, so the T cell is sort of stimulating itself and its neighbors, and the neighbors are stimulating themselves and this T cell. So this is both autocrine and paracrine signaling. And so this is sort of how it works with the T cell making both the IL-2 and the receptor. As I learned quite a bit during my PhD, IL-2 also plays a pretty important role in the contraction phase. And contraction, as I said, is a really important part of the life of a T cell. And so there's one other piece I want to tell you about in terms of contraction right now. You guys know of a few different types of cell death. We talked about pyroptosis earlier in the semester, and pyroptosis is a programmed form of necrosis. There are also non-programmed forms of necrosis. And you know a lot from Dr. Dunaway's class about apoptosis. The contraction phase of T cells happens via apoptosis, and you can see an apoptotic cell here where a chromosome is condensing and we're getting little membrane vesicles compared to our nice healthy cell. Contracting cells contract via the process of apoptosis through signaling through proteins known as Fas and Fas ligand. Our T cell that is about to die has a protein called Fas on its surface. Fas binds to Fas ligand. We can have fascinating conversations about where the Fas ligand comes from in the case of contraction. I'm leaving it out right now, but if you really want to know about it, I can tell you. It is a whole really cool thing. It's just kind of tricky. Fas, when it binds to Fas ligand, makes a trimer. This binds to a signaling protein known as FAD. And FAD can activate a protein called procaspase 8 to become active and be caspase 8. Caspase 8 can turn on some things in terms of mitochondrial apoptosis, but caspase 8 can also just directly cleave caspase 3, which is the bottom, the executioner caspase, and turn apoptosis on right there, not even requiring the mitochondria. So as soon as our cell that's getting ready to contract has Fas on its surface, it can get this interaction and be pushed into apoptosis, and it's pretty quick because caspase 8 is directly able to turn on caspase 3. Yes, Molly?
You might have already said it and I missed it, so sorry, but where does the Fas ligand come from?
So the Fas ligand, in the case of T-cell contraction, comes also, please don't be confused when I say this, also comes from the T-cell. So the T-cell actually bends back on itself and kills itself. I left that part out, but that's actually, the T-cell actually bends around and kills itself by having the Fas ligand as well. So, and just so you are aware, there is a very severe autoimmune disease that happen in people who have mutations in either Fas or Fas ligand. So if people are missing Fas or Fas ligand, they have an autoimmune disease known as ALPS, or autoimmune lymphoproliferative syndrome. And so in this little girl, you can see a good one right here. All her lymph nodes are permanently swollen because her T cells cannot contract. Basically, her T cells divide and divide and divide and get activated, but never undergo contraction. So all of her lymph nodes, there's more, like this is her lymph node on her back, which you can't see super well here. But all of her lymph nodes are really, really, really swollen. And these patients end up having really bad autoimmunity and sort of general immunopathology because that contraction cannot happen of their T cells. So this contraction phase is incredibly important for our T cells biology. So all of these events, the signal 1, the signal 2, the cytokine production, the proliferation, etc., are all really important to help our cell go from being a naive cell to a cell that can now exert its effector function. It can do its job. For the rest of the time today, I want to tell you about that job of CD8 T cells, and then we'll do CD4s on Friday, and whatever I don't finish about CD8s today. So when a CD8 positive T cell has finally become activated and differentiated, it can now be called a cytotoxic T cell or a cytotoxic T lymphocyte, a CTL. CTLs are able to perform their effector function in tissues. So at the beginning, our cell is a CD8 cell. It gets activated by an antigen-presenting cell, like a dendritic cell in the secondary lymphoid organ. It proliferates and differentiates, and then when that same cell goes out to tissues to find the infected cells, that cell will kill upon TCR interaction. It didn't kill here, but it did kill here. So our cell has changed its abilities. One thing that you can notice is that our cell is killing in a specific fashion. It's killing this infected cell and leaving the neighboring cells untouched. And you can also see this process happening here. So the cells in this video, we've got some T cells, we've got some cancer cells, and there is a dye in the media. The dye changes color when it gets inside of the cells. So at the beginning, our cells are able to exclude the dye stays outside, so the cells don't have a color. If the cells start to undergo cell death, then the dye can leak into them, and you'll see a color change. And so you can watch our cytotoxic T cells interacting with our tumor cells. The cytotoxic T cells are the little circly ones. The tumor cells are the big stretchy ones. You can see the interaction. You can see that the T cell lives through it and leaves and goes on somewhere else. It might go touch the other tumor cell and kill it. And you can see this whole process here. So you can see that our cytotoxic T cells are pretty effective. They're able to interact nicely with an infected cell and then leave that process unscathed, while the infected cell or tumor cell in this case does not fare quite as happily. And it's a pretty specific interaction. So our T cell interacts with the tumor cell, and then boom, that cell is compromised. That cell undergoes apoptosis. And so this is the job of an activated CD8 positive T cell. So our CTL is able to recognize antigen that is presented on MHC class 1 and kill that infected cell. It will program that cell to die and then leave, and our CTL is able to kill multiple cells one after another. It's not like they're one-time use only types of things. So you might wonder, well, okay, so how does the T cell do its killing job? And there are two big ways that CTLs are able to kill their targets. One of them is with FasL, which is why I told you about FasL already. So CTLs have FasL ligand on their surface, and infected cells can have FasL on the surface, and this FasL ligand can interact with FasL on the target cell to induce apoptosis of the target cell. Exactly the same way that you saw FasL signaling leading to death in the case of the cell undergoing contraction. The other way that this can happen is through something known as cytotoxic granule release. So our CTLs have granules in them that can be secreted. One thing that's and these granules can include some toxic molecules that are able to kill the target cell. And we'll talk about what these molecules are and how they kill on the next slide. But one other thing that's really, really cool about all of this is that the cell actually performs directional secretion. So the cell does not take its granules and secrete them in 360 degrees around the whole cell, because then they might kill the infected cell and all the neighbors. They would really destroy that area. So in fact, our cytotoxic T cell orients its entire set of organelles, is really its entire cytoplasm, so that the granules are all right next to the target cell to be killed, and puts integrins to be a little wall, so that the cytotoxic molecules can only go in the cytotoxic synapse and not diffuse randomly outside of this area in order to kill other things. And so this is kind of why that ring, that target ring that we talked about last time, is so important, because it also forms a ring that allows these secreted products to stay in one place and only go towards the cell that we are trying to kill. And so you can see that in cartoon form here, but you can also see that in the microscopy image here, how all of our cytotoxic granules are reorganized right towards the contact site to be secreted directly at our cytotoxic cell. Your textbook shows this process here, where our cytotoxic T cell interacts with the target cell. Within about a minute, reorganization of that CTL starts to happen, so the pink dots start to move closer and closer to the target cell, so they're all right next to the target cell. Those are then exocytosed right at the target cell. The TTL then leaves to go kill somebody else, and within about 30 minutes, that target cell is going to undergo apoptosis. So I find the cell biology of the cytoplasmic reorganization super fascinating here. So then, of course, you might wonder, well, what is in this granule? What are these cytotoxic things? There are a lot of answers. There are a lot of things that are in these granules, but I'm going to tell you about two that are particularly important. One of the two is known as perforin. If you see the word perforin, what do you think of? Yes, Mel.
Perforating.
You think of perforating. What's perforating mean? Poking holes. Guess what perforin does? Pokes holes. It perforates the cell. So perforin is in those cytotoxic granules as monomers. When it interacts with the target cell membrane, it's able to polymerize into pores and perforate the cell membrane, and thus cause holes in the membrane of the target cell, potentially leading to the apoptosis of that cell. If you actually look at the structures, the perforin structure is really similar to the C9 complement structure. The pores look super similar, and in fact, some textbooks totally lie, and they show pictures like this, and they don't really know if they're perforin or if they're C9, and they just tell you they're one or the other. Perforin will give us holes in the target cell, which can lead to things like osmotic lysis of that target cell. But perforin can also let other things in that granule into the cell. So it also forms a way that some of the other cytotoxic components make it into the cell. The most famous one that I'm going to tell you about is known as Granzyme B. So Granzyme B is able to go through perforin's pore. The location of the pore, we've changed in the field where we say the pore is about 50 times, and that's since I've been a grad student working on this. It gets changed like every 5 minutes. So this version shows it slightly differently than the other figure, even though they're next to each other in your textbook. It's okay, don't worry about it. I think it happens at the cell membrane, whatever. Anyway, wherever perforin makes its pore, that eventually lets Granzyme B come into the cell. Granzyme, you might guess, since it has zyme in its name, is an enzyme. It's an enzyme in the granule, so it's granzyme. Granzyme is able to cleave a number of substrates. One of them is BID to turn on the mitochondrial apoptosis pathway. The other is, again, caspase 3 to turn on apoptosis without even worrying about the mitochondria. So again, we pretty rapidly turn on apoptosis as a result of Granzyme B release. And so all in all, our CTLs are able to exert their effector function in a bunch of ways. They might use Fas and Fas ligand. They also might use their cytotoxic molecules like perforin and granzymes. There are others. They also make a lot of a cytokine called interferon gamma, which can be very important in helping to deal with those effector cells. So I heart CD8 T cells. I've worked on CD8 T cells quite a bit. But the one other thing to tell you about CD8 T cells is if we want to make a CD8 T cell into a CTL, there is one other important thing we need. And that is some help from CD4 cells. So CD8 T cells require some cytokines from CD4 T cells. There are a few different models of this which are shown differentially here, but the moral of the story either way is that the CD8 T cell in purple has to get some cytokines that originally came through a process of CD4s. And so if we don't have CD4 help, we don't have CD4 cells, we actually can't make a good CD8 response, which is why we also have to fully understand the CD4 response, which is our job